Okay, this mini lecture is to talk about enzyme inhibitors. So we've talked about enzymes before, and most enzymes are proteins. They have a specific shape that gives them function. Enzymes are called catalyses, so it's a catalyst, which means it speeds up a reaction, and enzymes are, enzymes are reused. So remember, they do their function, they bind reactants, release products, and can be used over and over again. Here we go. The part that's really important for shape and function is the active site. So the active site is where the chemical reaction happens. And remember that this is very specific. So one enzyme will catalyze one type of reaction, and it's because that active site has a very specific shape to bind the substrate. So the active site is where the substrate binds. Okay, so this little diagram on the first slide is showing you overall what we're going to be talking about. We're going to be talking about both competitive and allosteric inhibitors. So let's look at a competitive inhibitor. So an inhibitor, let's go back for a second. Let's write, well, what an inhibitor is basically going to block the enzyme function so that the enzyme cannot make the chemical reaction happen. And the way it's going to block the enzyme function is, let's say, put by interfering with the active site. And so you're going to see that there's two different ways. You can just see this by where the inhibitor is binding. Two different ways for inhibitors to block enzyme function by interfering with the active site. Okay, so now let's talk about a competitive inhibitor. A competitive inhibitor binds to the active site and blocks, like physically blocks, the substrate from binding. So that is how a competitive inhibitor inhibits the enzyme, because here's the normal reaction. We have a substrate, and you can see specificity, right? It has to fit right there, and it binds to the active site, and the reaction happens. So with a competitive inhibitor, the inhibitor actually has a very similar shape to the substrate. So the inhibitor binds to the active site, and that blocks the substrate. So now the substrate can't bind, and you don't get the chemical reaction happening. Okay? So competitive inhibitors compete for active site binding, and by blocking the ability of the substrate to interact with the enzyme, no chemical reaction happens. That's different than a non-competitive or allosteric. Okay? Allosteric is talking about shape. So an allosteric binds somewhere else okay, let's write. Binds to the enzyme at a location other than the active site and an allosteric prevents the enzyme from working by changing the active site shape. So, an allosteric inhibitor binds to the enzyme at a location other than the active site, so we have it binding here, and changes the active site shape so it can no longer bind the substrate. Okay, so we're really focusing on this half of the diagram. I'll talk about the left half in just a second. Okay, so an allosteric inhibitor is going to bind somewhere on that enzyme, and it's changing the active site. So now you can see that that little diamond shape is not going to fit into the active site. It says wrong conformation, and that equals no chemical reaction. Okay. So, competitive inhibitors bind to the active site and physically block the substrate from binding, and that results in no chemical reaction. Okay. And allosteric inhibitors bind somewhere else, change the active site shape so that it can no longer bind the substrate, and there's no chemical reaction. Now, there are also allosteric activators, and this is not what you need to know, so I'm just giving you this as an aside because it's talked about in books. An allosteric activator, you could figure this out because you know allosteric means a different site. An activator is something kind of opposite of an inhibitor, right? So it activates, it promotes it. So an allosteric activator will actually bind to the enzyme and make the active site functional. Okay. So an allosteric activator activates the enzyme. So the enzyme might be sitting without the proper active shape because it's waiting for a signal from the cell that says, okay, we need you now. And that would be an allosteric activator. So all these become really important when you look at cell biology and physiology and the actions of enzymes. So we're just covering some basics for terminology. And you need to know the difference between a competitive and an allosteric inhibitor. All right, thanks. Oh, I was wondering if I had a summary slide. Okay, so this is just another image. I like to give lots of images. I was afraid that if I went to the slide and it was done, it would cut off my recording. So I summarized, but here we go. Normal reaction, substrate binds the active site, you get products, you get the enzyme back. Competitive inhibitor binds the active site, substrate binding is blocked. Non-competitive inhibitor changes the active site. This should be, no, I don't like this. Substrate can bind, but reaction's blocked, or substrate can't bind at all. I should have looked at this figure a little bit better. But the key is this inhibitor binds to the active site. A non-competitive or allosteric inhibitor binds somewhere else. Both are blocking the reaction from happening. Okay, now I'm done.